Please join me now in welcoming Cassandra Griffin, the Coordinator of Biobanking and Donation, and she really is the cornerstone of our research program.
First time I've been called a cornerstone, so thank you very, very much. That seems substantially large shoes to fill, particularly when I then nearly stack it on the steps. But hi everyone, so my name's Cass and I'm your friendly neighbourhood biobank manager. A lot of you I would have spoken to over the phone over the last couple of years. I generally try to get out and meet people in person, but it's been quite difficult for obvious reasons over the last couple of years. So before I do get started, for anybody who is contributing to the biobank, who receives our requests for bloods, thank you so much for what you're doing, and please pop over and say hi, because I won't be leaving straight away afterwards. I would love to be able to put some faces to names, and I've also got some updates on the type of research that your samples is contributing to, so it would be great to share that with you and get to know you all. All right, please work. Huzzah. Okay, great success. So, as Professor Ackland just said, one of the hugest hurdles that we face in brain cancer research is that, relatively speaking, we don't have a complete understanding of the neurobiology of brain tumours. There are so many unanswered questions, and there are a number of factors that contribute to that. But one of the key factors that contributes is that we don't have the availability of tissues that we have from some other disorders. In 2014, our service, which at the time was known as Hunter Cancer Biobank, we've now rebranded as the Mark Hughes Foundation Brain Bank, partnered with the Mark Hughes Foundation to set up the Mark Hughes Brain Bank, and it's a collection of surgical tissues and of blood samples. So what is a biobank? I don't know if anybody's encountered a biobank in any other aspect of their life, but biobanks exist for all sorts of biological organisms. There are biobanks for seeds and for native flora. There are animal biobanks, everything from shark eyeballs to penguin poo. If it's a biological organism, we can bank it and we can use it for research. What we bank up at HMRI are solid tissue, blood, other body fluids and their derivatives, just about anything we can get or that people are willing to donate. And biobanks allow what we call translational research. So this is moving from laboratory-based models of research, where we're looking at cells and DNA and those very small molecules, moving them into people. So it provides that stepping stone and the bridge so that we can get new developments and new research to you faster. So the Mark Hughes Foundation Brain Bank is by definition a patient-driven biobank. All biobanks are reliant on donors and contributors, and all good biobanks should be driven and informed by their community. But the origin stories of our biobank are particularly special and really do define a patient-driven program. So in terms of our brain cancer biobanking, many of you will be familiar with this story because you will have been through the process. But when patients are diagnosed, many are offered the opportunity to participate when they first meet with the brain care coordinators. When you have tissue removed during surgery, that will go up to a pathologist who will process it in certain ways, and I'll speak about that in a minute, and then submit that for it to be assessed by a pathologist to make a diagnosis. It doesn't happen all that frequently with brain tumours because quite often the tissue that's removed is quite small. But if you think about a breast cancer or a colorectal cancer where quite a large piece of tissue is removed, what they need for diagnosis versus what's actually removed is often a huge difference. And all of that excess tissue used to be thrown away. What we do in the biobank is we take anything that's not absolutely necessary for diagnosis, we put it into a formalin block and we bank it. And that's what we provide to researchers then for use. We also collect serial blood collections. Serial blood collections are taken at various time points throughout a patient's journey so that we can look at how the tumour changes, how it responds to treatment, and where those key steps are that maybe we need to be zoning in on for our research priorities. My animation worked. Again, great success. So here is a piece of tissue that's been removed during surgery and it's being fixed in formalin. So what that does is it keeps all of the cellular structure and all of the histological structure as close to how it would be when it was still inside a person. So it keeps it looking the way that it should. It sits in formalin usually for around 24 hours before it gets processed. When tissues are processed, they're dehydrated, they go through different temperature cycles, and again, that's about fixing it and trying to make it a stable piece of tissue. It then gets embedded in wax or paraffin, so it creates a tiny little block that looks like this. That block then can be sectioned by a microtome to produce a 4-micron section, so thinner than a human hair, which we mount on a slide, and we then use various stains to interrogate it and investigate it so that researchers can look for different proteins or different cellular structures that might be able to answer some of our questions about brain cancer. And I've got some photos down on our booth if you'd like to come and have a look of different things that we've stained for. In terms of blood samples, so when we send out a request to people for a blood collection, we collect 2 tubes. One is an EDTA and one is the funky looking camo-topped one, which is known as a Streck tube. From these 2 tubes, we're able to produce all of these products, and they're banked in tiny little aliquot tubes. And again, I've got an example down there to show you just how much comes from a single blood donation. And quite often researchers only need 1 aliquot for the work that they're doing. So from that one collection, we can produce all of this. And again, it's of huge benefit to researchers. Okay, so 2 years after we established the sequential blood program, an extremely special patient, and her daughter is here today, contacted us and said to us that she would like to do more and that she would like to donate her brain to brain cancer research so that we could keep working towards finding a cure. This wasn't something that we had done before, and it was definitely new ground for the Biobank. So we worked very closely with the Mark Hughes Foundation, with her family, and with the healthcare providers around us to see if we could get this happening. There are brain donation programs in Australia and certainly in New South Wales, but the vast majority of these have been set up to look at neurodegenerative disorders and things like Parkinson's disease or Alzheimer's disease, other types of illness where the brain might lose mass or where the neurons start to change in different ways, but none where the brain effectively expands or where you have a mass creating pressure. And because of that, the conditions that we need to be able to facilitate something like this are very different to those that we would set up for an Alzheimer's or a Parkinson's patient. So we tried to set up something that would uniquely fit the criteria of brain cancer so that her donation would have the maximum benefit possible for research. And we managed to do that. And what we have set up is a program that is completed within 4 hours from the time that a person passes away until the time that that tissue is in the laboratory. And everything that we do, all of the steps that we take and all of the decisions that we've made are uniquely informed by what we understand about the pathophysiology or the way that the disease is caused of brain cancer. We're also doing a lot of research now, working with the families of people who have passed away and donated their brains to understand the impact that that has on them to be able to make that decision, and the impact that it has on people afterwards, knowing that their loved ones have a legacy, so that any benefit from this program can be really felt by the community. These are some of the people who have donated. Since Jackie, we've had 24 people who have donated to the Brain Bank, all absolutely remarkable, beautiful, wonderful people who I'm extremely honoured to have met. More than 50% of the grants that our biobank has serviced, so researchers will apply from grants for different bodies, be they philanthropic bodies, government bodies, all sorts of people. And we work across all cancers. We also work across multiple sclerosis, diabetes, preterm birth, maternal health, lots of different areas. But at the moment, more than 50% of the grants that are awarded to the researchers we collaborate with have been in brain cancer and have utilised samples that have been donated to the Mark Hughes Brain Bank, which we think is an absolutely awesome achievement. And our goal is to keep doing this and keep working with the community to make sure that our researchers have a world-class resource right here in Newcastle to keep moving forward and to try and make a difference to people who are living with brain cancer. This gives you an idea of sort of the quantity of samples that are being sent out. So these are the surgical samples that we brought in last year. This is last year's samples, and obviously things were a little bit slower last year due to COVID and due to our inability to interact with patients as closely as we normally would. But in terms of quantities, you can see the samples that come in a year. versus the number of samples that are being sent out in a year. So my motto, managing the biobank, has always been that the best biobank is an empty biobank. Samples should be going straight off the shelf and straight out into research where they belong and where they can do the most benefit. And that's exactly what we're working towards and exactly what we intend to keep doing. So before I finish up, I just wanted to give you a little bit of an overview of some of the research projects that are ongoing in Newcastle. Our samples have been sent all over the world, but these are the ones that are specifically going on our doorstep. And again, I've got a booklet down here if anybody's interested that can give you some more detail on the projects that are ongoing. In terms of the research that's going on, we know it doesn't happen in a bubble, and our researchers have wonderful networks and collaborators all over the world, and they share samples and they share ideas, and this is the only way we're going to be able to move forward and find a cure. Some of the projects that are ongoing, we have teams working on blood tests that may be able to monitor progression and guide treatment decisions without the need for other more invasive procedures. We have researchers doing a lot of work on the tumour microenvironment. So the microenvironment is everything that goes on around the tumour. It's where the tumour lives, the cells, the growth factors, the nerves, the blood vessels, everything that influences the way that it might behave. We have researchers looking at the side effects of temozolomide, and I'm sure that unfortunately many of you will be familiar with some of these side effects. And for some people, the side effects of temozolomide mean that they can't continue with longer treatment. So these researchers are working hard to understand who might have unfortunate side effects, how we might be able to navigate these, and trying to predict it so that people don't have to experience that. Another huge area of research is looking at tumour recurrences, trying to understand why tumours come back and how we can prevent that and how they're different when they do come back. Unlocking the genetic code, and again we have a wonderful genomics facility here in Newcastle up at HMRI, and we're gradually working through the entire brain bank of samples, sequencing tumours to try and map out their genetic code and see what might be similar, what might be different, and what might provide new opportunities for targets. And lastly, we have a group over at Callaghan who are working on nerve tumour crosstalk, looking to find ways that we might be able to harness nerves and stop some of those signals that tell the tumour to grow and tell the tumour to be aggressive. So this is the wonderful team that I get to work with, and supporting MHF is a family affair in my household. That's my pup proudly sporting her Mark Hughes bow tie. But really, I just want to say thank you to all of you, because this program does not exist without you. It was inspired by you, it is driven by you, and it will only continue because people like you won't stop fighting. So thank you, and I really hope that very soon I'll be up here with some big news from the samples that are stored in our bank.